Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Diversity of Plants Zone. In this channel, you can get every information which is related to the study and diversity of the plants throughout the world. Before starting this important topic, I request you all to please subscribe to my YouTube channel and like and share as well. Today's topic of my video is about the domain Eubacteria. I hope you will enjoy this video. So let's start the video. First of all, I will discuss about the Eubacteria. So what is meant by Eubacteria? Eubacteria are prokaryotic microorganisms consisting of a single cell, lacking a nucleus and containing DNA. Is a single circular chromosome. Eubacteria can either be gram-negative or gram-positive. They have economic, agricultural, and medical importance. They include E. coli and Lactobacilli and Azospirillum. So this is the structure of the Eubacteria as shown into the figure. In its structure, it includes pilus. This is pilus. This is capsule. And this is cell wall. The next is the plasma membrane. These are plasmids. And this is flagellae. These are ribosomes. And you can see the nucleoid region, which is also known as the DNA. And this is the cytoplasm. So Eubacteria is also known as true bacteria. Bacteria excluding the Archaea. This is the structure of Eubacteria as shown into the figure. So next I am going to discuss about the Eubacteria definition. Eubacteria, biology definition, literally means true bacteria. They include all bacteria except for Archaebacteria, and these bacteria form the domain Bacteria, previously domain Eubacteria. It is one of the three domain systems of classification. The other two are domain Archaebacteria, which is now known as Archaea, and the domain Eukarya, which is now known as the eukaryotes. So Eubacteria are prokaryotic organisms, for example, lacking a membrane-bound nucleus and predominantly unicellular and with DNA in a single circular chromosome. Cell wall when present is made up of peptidoglycan, and examples are E. coli and Staphylococcus, Salmonella and Lactobacillus. And synonym is true bacteria. Or Eubacteria or true bacteria are single-celled prokaryotic microorganisms that have a range of characteristics and are found in various conditions throughout all parts of the world. All types of bacteria fall under this title except for Archaebacteria. So since Eubacteria are so common, this group comprises one of the three domains of life, which is called as bacteria. So this is again the structure of the Eubacteria as shown into the figure. In its structure, it includes the flagella. So this is capsule. This is cell wall. This is plasma membrane. And the next is plasmid DNA. So this is cytoplasm. These are fimbriae. This is chromosomal DNA, and these are food granules, and these are ribosomes. So Eubacteria, this is bacterial cell diagram with label as shown into the figure. Next I am going to discuss about introduction to Eubacteria. All living organisms are classified into three major domains. And the first is the domain Eukaryota, which includes all eukaryotes. Second is the domain Eubacteria, that includes true bacteria. And third is the domain Archaea, which includes Archaebacteria. So domain Eubacteria includes the true bacteria, and it is one of the largest domain that includes a large group of the organisms. Now the question is, so what is Eubacteria's cell type? Eubacteria as well as Archaebacteria are prokaryotes. And conversely, the Eukarya is composed only of eukaryotes. Unlike simple prokaryotic cells, the eukaryotic cells tend to be more complex. They include the unicellular and multicellular protists, plants, algae, and animals. Now the next question is, do prokaryotes have cell walls? Prokaryotic eubacteria includes the bacteria with cell walls consisting of peptidoglycan. However, not all bacteria have cell walls, but all eubacteria have cell membrane. Bacteria's cell membrane consists of the glycerol and fatty acid which are combined together by an ester bond. Now the next question is, what are eubacteria? Eubacteria, sometimes called simply as bacteria, are small organisms that cannot be seen by the naked eyes. Thus microscopes are used to visualize and study their morphology. To do so, bacteria are stained. And staining is an essential microbiological technique as it helps in highlighting the whole bacterial structure and cellular shape. And bacteria are classified according to gram staining, and some of them are considered as gram negative, while the others are considered as gram positive. Gram positive bacteria have several layers of peptidoglycan in their rigid and thick cell wall, which is highlighted by gram staining. The cell wall of gram-positive bacteria contains the lipoteichoic acid that is composed mainly of alcohol and phosphate in the plasmic space. So lipoteichoic acid function helps the bacterial cell to grow. It also protects the bacterial cell wall from lysis. And lipoteichoic acid provides the wall antigenic specificity to gram-positive bacteria. Therefore, it helps in the grouping of gram-positive bacteria into several types. The gram-negative bacterial cell wall consists of only one layer of peptidoglycan and has no lipoteichoic acid, so gram stain cannot bind to their cell wall. As a result of its weak structure, the gram-negative bacterial cell wall is susceptible to mechanical damage. The outer membrane of gram-negative bacterial cell wall consists of negatively charged particles such as lipopolysaccharides, phospholipids, and lipoproteins that enable the gram-negative bacteria to evade immune cell detection and phagocytosis and the action of the complements produced by the immune system of the host. Moreover, the outer structure protects the gram-negative bacteria from bile salts, heavy metals, digestive enzymes, detergents, and some antibiotics such as the penicillins. So, this is gram-positive versus gram-negative. So you can see in the structure of gram-positive bacteria, this is lipoteichoic acid. And in blue color you can see that this is the peptidoglycan cell wall. This is plasma membrane. And this is cytoplasm as shown in the figure. On the other hand, in the structure of gram-negative bacteria, you can easily see that these are porins. And this is lipopolysaccharide in green color. This is outer membrane. And this is peptidoglycan cell wall, you can see here. And this is the lipoprotein in purple color. And this is the periplasmic space. On the other hand, this is inner membrane and this is plasma membrane. And in plasma membrane you can see easily these are membrane proteins in light blue color. And this is phospholipid in orange color. So this is gram positive versus gram negative as shown into the figure. Now the next question is, how do bacteria reproduce? Eubacteria reproduction usually includes dividing the parent cell into two daughter cells after the replication of genetic material in a process which is called binary fission. Some bacteria have ability to form a spore in unfavorable conditions such as the deficiency of nutrients and exposure to chemicals or radiation. And these spores cannot reproduce, however, they are highly resistant to toxins, radiation, heat and dryness. Spore forming bacteria such as Bacillus and Clostridium are considered to be virulent bacteria, so the sterilization techniques must eliminate the bacterial spores. When environmental conditions become favorable again, the bacterial spores start to vegetate and reproduce again. So this is the question, how the eubacteria reproduce by binary fission. This is plasma membrane, this is chromosome and this is cell wall of the eubacteria. Now the chromosomal replication first takes place. After chromosomal replication, there is a step of septation or constriction. And after septation and constriction, the cell is divided into two daughter cells as shown into the figure. So you can easily see that how eubacteria reproduce by binary fission in this figure. Next I am going to discuss about eubacteria characteristics. Eubacteria characteristics. Now the question is, are eubacteria prokaryotic or eukaryotic? And what are the three characteristics of eubacteria? Eubacteria are unicellular prokaryotic cells. They contain a circular chromosome. Moreover, the eubacterial cell wall is composed of peptidoglycan and they greatly differ in terms of morphology or physiology. And the next question is, what is a eubacterial cell structure? So eubacterial cell type is prokaryotic. Their cells lack nucleus and cell organelles present in eukaryotic cells. Their DNA is not inside a nucleus. And these structural differences between prokaryotes and eukaryotes are greatly important since the ability of the antimicrobials to eliminate the bacteria depends mainly on targeting these differences, making them selective to bacterial cells without affecting the eukaryotic human cells. Now the next question is, are eubacteria multicellular? The size of bacteria ranges from 0.2 to more than 50 micrometers and bacteria are usually unicellular. However, some colonies of bacteria are found as filaments or aggregates in the form of surface biofilms. So bacteria are unicellular prokaryotic microorganisms. Their cells contain carbohydrates, lipids, proteins and nucleic acids, and they produce energy and metabolites using the different chemicals since their cells lack the cellular organelles such as the chloroplasts, nuclei and mitochondria that perform these functions. Usually the bacterial chromosome is circular, however some species have two chromosomes like the Vibrio cholera, while the other species have a linear chromosome. So DNA of these cells is not linked with a histone, rather it is linked to different proteins. Bacterial cells are cylindrical, spiral, spherical or pleomorphic, irregular in shape. Some bacteria obtain their energy from sunlight and use carbon dioxide for obtaining carbon to perform photosynthesis to create the cellular biomass. These bacteria are called as photosynthetic bacteria. Among these species are Cyanobacteria, which is also known as the blue-green algae. Other eubacteria species get energy through organic and inorganic material metabolism, such as from sulfur and ammonia. So eubacteria are mostly heterotrophs, which take food from an outer source. So most heterotrophs decompose dead material or parasites that live on or in a host. Other eubacteria are autotrophs by making their own food, and they are either chemosynthetic or photosynthetic. The most important autotrophic eubacteria are cyanobacteria. Respiration in eubacteria is either aerobic or anaerobic. So anaerobic bacteria undergo fermentation as an example of respiration. So this is a diagram of typical cyanobacteria cell showing the parts. So it includes ribosomes. These are ribosomes, and this is nucleoid DNA ring, and these are thylakoids. These are thylakoids, and this is rubisco. This is rubisco, and the next is the carboxysome. This is carboxysome, and so the cell wall includes cell membrane, peptidoglycan layer, and this is outer membrane. And the next is the mucoid sheath. This is capsule, and this is the slime coat, and this is the thylakoid membrane as shown into the figure, and the next is the phycobilisome as shown into the figure. So this is the whole diagram of a typical cyanobacterial cell which is showing its different parts as shown into the figure. Eubacteria are microorganisms lacking a defined membrane nucleus have several general characteristics. As prokaryotes, they do not have any membrane-bound organelles. Most eubacteria are enclosed by a cellular wall, which is made up of peptidoglycans in a cross-linked chain pattern. This gives the wall of the bacteria the strength needed to maintain its shape and size through changing environments. Small molecules can diffuse through the cell wall, but larger molecules and ions need carrier proteins and channel proteins to enter the cell. So this is again the structure of a typical eubacteria, and it includes the pilus. This is capsule, this is cell wall, this is plasma membrane, this is plasmid, and this is flagellae. These are ribosomes, and this is nucleoid DNA, and this is cytoplasm. So this is bacterial cell anatomy as shown into the figure. Some bacteria may have flagellum, a structure which is composed of a protein filament, so that is used for movement. Or other bacteria have pili, which are small projections all over the outside of the cell that are used for sticking to surfaces and transferring DNA. So when a large number of bacteria are attached to the surface and surrounded by a polysaccharide sac, this is referred to as a biofilm. This complex has a antimicrobial resistance. The fluid contained by a plasma membrane of the bacteria is the cytosol. It is composed of mainly water, approximately 80%, but it has a gel-like consistency because it is filled with dissolved nutrients, cytoskeletal elements, DNA and other substances. So eubacteria do have ribosomes that are organelles that are composed of RNA and protein that complete the process of protein translation. So bacterial DNA floats freely within the cytosol, though it is concentrated in the nucleoid. Bacterial chromosomes are often circular but can also be linear in shape. So this shape comes in handy when a bacterium is undergoing replication. So bacteria can asexually reproduce through binder fission or budding. And bacteria that undergo binder fission must first elongate and duplicate their DNA before separating into two new and similarly sized cells. Budding happens when a parent cell produces a small extension of the cell membrane and fills it with the duplicated DNA. On the other hand, the binder fission produces two equal daughter cells, while the budding process a small new cell while the parent cell remains quite large. So these are the stages of the endospore formation. So eubacteria have the ability to form tough endospores to survive under unfavorable conditions as shown into the figure. So during times of extreme conditions and not conducive to replication such as the starvation, eubacteria have ability to become endospores. In this state, the bacteria can tolerate exceedingly high and low temperatures, acidic and basic conditions, and large amounts of radiations. So endospores are extremely hard to kill. Surprisingly, they can be boiled for hours and still survive. Endospores can only be made by the gram-positive bacteria. Within the endospore remains the bacterial DNA, but the cytosol loses water and becomes extremely concentrated. And this is thought to help in protecting against high heat. The bacteria will take on a tough coating composed of calcium and dipicolinic acid, creating a dense and impregnable barrier to stabilize the DNA within the cell. And DNA repair enzymes are so still active, adding in the resistance of the endospore. So plasmids are also found within the bacteria and separate from the bacteria's circular DNA, also referred to as the replicons. Plasmids are autonomous replicating DNA molecules. Essentially, these small rings of DNA allow the bacteria to transfer genes between organisms, and these elements allow for the horizontal gene transfer, which is a way for a bacterium to gain new genes and traits such as antibiotic resistance. They primarily add in the rapid mutation in bacteria to changing environmental conditions. Similar to the other genetic material, the plasmids can be passed on to daughter cells during the replication. So this is a 3D illustration of a colony of eubacteria as shown into the figure. Next I am going to discuss about the major characteristics of eubacteria. So major characteristics of eubacteria, first it includes the metabolism. Because of the diversity of species classified under the domain Eubacteria and the fact they are found in a wide range of habitats, and they have been shown to display a higher diversity in their energetic sources and metabolic pathways and when compared to eukaryotes. In the absence of organic matter, many species described as chemoautotrophic eubacteria have been shown to obtain their energy through the oxidation of mineral compounds. A good example of this is the bacterial species that oxidize the nitrates or methane In the process, they also play an important role in converting the various material into usable compounds. Using such compounds as ammonia and nitrifying bacteria produce the nitrates that are used by plants to make the proteins and amino acids, etc. Some of the main nutritional modes of different species in the domain Eubacteria include autotrophic. Autotrophic produce their own food through photosynthetic process. On the other hand, heterotrophic obtain nutrients from their environment because they are unable to synthesize their own food. And next is the strictly or facultatively aerobic, survive in the presence of oxygen, which are strict aerobes, or can switch to the anaerobic respiration in the absence of oxygen. So these are considered as facultative anaerobes. Next is strictly or facultatively anaerobic, survive in the absence of oxygen. So these are strict anaerobes, or can survive with or without oxygen. So these are facultative anaerobes. For eubacteria, diversity in the modes of metabolism is also due to a wide variety of chemical compounds that serve as substrates, as well as the ability to produce energy in the different environmental conditions. Whereas various photosynthetic forms require the oxygen for photosynthesis, others can depend on fermentation process for the energy and only grow well in the absence of oxygen. In the food chain, they play an important and various environments. They have important role in various environments, not only as prey for various unicellular eukaryotes, but also as producers that spore the various metazoans and decomposers that break down dead material that is then reused by the plants. So next is the main characteristic, which is cell structure. All eubacteria species have a cell wall consisting of peptidoglycan. However, according to various studies, species that lack this structure are those that have lost it secondarily. So peptidoglycan is made up of glycose, aminoglycan, which in turn consist of alternating residues of D, glucosamine, and moramic acid. So during peptidoglycan synthesis, the carboxyl group of the moramic acid is typically substituted by a peptide consisting of L and D amino acid residues. So depending on the organism, these peptide units may be cross-linked through peptide bonds to produce a giant macromolecule, and that contributes to the cell wall. In gram-negative species, the macromolecule occurs as a monomolecular layer which is located between the inner and outer membrane. In gram-positive bacteria, it appears as a multimolecular layer, covalently or non-covalently bound to such compounds as teichoic acid or neutral polysaccharides. So typically, bacteria that lack a cell wall is given the suffix plasma. Some examples include ureaplasma and mycoplasma. These characteristics allow them to remain resistant to various antibiotics that function by targeting the cell wall structure. In gram-negative bacteria, the peptidoglycan layer make up about 80% of the cell wall. So the next major characteristic is the reproduction. While some species have been shown to exchange the genetic material through cell contact, asexual reproduction is the main mode of reproduction in eubacteria. So some of the main methods of asexual reproduction in eubacteria include binary fission. So this is the most common mode of reproduction in eubacteria, and during favorable environmental conditions. During binary fission, genetic material of the organism or nucleoid divides without mitosis. This occurs through DNA replication, a process that produces two strands of DNA. DNA replication, which is bidirectional, originates from a given point known as the point of origin, producing the two circular DNA molecules. The cell divides into two when a peripheral ring of the membrane invaginates and grows towards the central part of the cell, thereby forming a double membranous septum. Cell wall materials are then deposited between the two membranes and causing the two cells to separate completely. And the next is Endospore Formation. For the most part, endospore formation occurs during unfavorable environmental conditions and is common among gram-positive and gram-positive bacteria and cyanobacteria. So during unfavorable environmental conditions, extreme temperature and presence of toxins or nutrient deficiency, etc., these organisms produce a thick wall to surround the cell to form the endospore. Before the cell is encased in the thick cell wall, the chromosome and the parts of the cytoplasm are also dehydrated. The cytoplasm and cell wall also deteriorate, giving way the thick wall that ultimately covers the cell. This makes the organism resistant to unfavorable conditions and allows it to survive for a long period of time, up to several decades. The thick cell wall covering the endospore consists of four main parts. And the first part is the exosporium, that is loose outer covering. Next is cortex. So cortex is the thick layer which is consisting of mucopeptides and carbohydrates. Next is the core wall. So core wall, also known as the inner cortex, and the core wall is largely composed of proteins. Next is the spore coat. Consists of disulfide rich proteins and resembles the keratin. So under favorable condition, the protoplasm swells up when it absorbs the water, allowing the dipicolinic acid to leak out. So this activates and causes the protoplasm to break the spore cover, and ultimately the bacterial cell emerges from the spore and continues developing and reproducing under favorable conditions. Some of the other common forms of asexual reproduction in Eubacteria include fragmentation, and the organism breaks into fragments and after growing to a given length as seen in Cyanobacteria. Next is budding. The organism produces buds that grow and detach to develop into new individuals. Next is the conidia formation, common in mycelial bacteria and involves the development of new individuals from conidia. So conidia are mostly rounded structures that cut from parts of bacteria. Sexual reproduction in Eubacteria involves an exchange of genetic material through conjugation, transduction and transformation. So next I am going to discuss about Eubacteria structure. Now the question is, are Eubacteria unicellular or multicellular, and how many cells do they have? The Eubacteria number of cells is only one. They are single prokaryotic cells and there is no such thing as eukaryotic bacteria. The structures found in Eubacteria cells are either external or internal to the cell wall. Structures external to the cell wall may be flagella, fimbriae and axial filaments and glycocalyx or pili. So each of these structures has its distinctive function, where some Eubacteria have flagella to facilitate their movement, and flagella are long filaments that facilitate the mobility of bacteria. So flagella consist of three main parts. A portion consisting of flagellin protein called the filament. For example, the long outermost part the middle hook, and the basal body that attaches the bacterial plasma membrane and cell wall. So fimbriae and pili are hair-like structures that are similar to the flagella but thinner. Pili or pilus are thin projections that are used for conjugation between the bacteria. For reproduction, after the bacteria are brought together by pili, the DNA moves from one cell to another, so new features as antibiotic resistance are transferred between cells. Glycocalyx surrounds some eubacterial cells, and it is a viscous polymer composed of polypeptides or polysaccharides and functions to protect the bacteria, so it is commonly known as the capsule. The capsule is considered to be one of the virulence factors of some bacteria, since it enables the bacteria to resist the phagocytosis by the immune system. So glycocalyx coat can also help the bacteria to attach firmly to the host cell, such as the Vibrio cholera that produces glycocalyx to attach to the intestinal cells. The cell wall of bacteria consists chiefly of a network which is peptidoglycan, so it may be associated with the other substances or present alone. The peptidoglycan network consists of disaccharide portions connected together by polypeptides, forming a lattice to protect the bacteria. So some antibiotics such as the penicillins and cephalosporins interfere with the structure of bacterial cell walls, leading to the cell lysis and rupture. Structures internal to the cell wall include cell membrane, cytoplasm, DNA, plasmid, and ribosomes. And like eukaryotic cells, the prokaryotic internal structures, such as those of eubacteria, are surrounded by cytoplasmic membrane consisting mainly of phospholipids. However, the cell membrane of prokaryotes is less rigid than that of many eukaryotes. That's because they lack the sterols, except for the mycoplasma that has sterol in its cell membrane. So some antimicrobial agents such as the quaternary ammonium and alcohol act by disrupting the cell membranes of bacteria. So additionally, a group of antibiotics that is called polymyxins can also damage plasma membranes, leading to the destruction of bacterial cells. The cytoplasm is a substance that is inside the plasma membrane and acts as a media for the internal structure of the cell. In eubacteria, the cytoplasm contains DNA that is not normally seen in the eukaryotic cytoplasm. Prokaryotic cells contain cytoskeletons in the cytoplasm that aids in the growth, reproduction, and maintenance of the cell's shape. The nucleoid DNA of prokaryotic eubacteria differs from that of eukaryotes. It consists of a long single circular double-stranded DNA which contains all the bacterial genetic information. The bacterial chromosome is not enveloped by a nuclear membrane like that of eukaryotes. It also lacks histone. Bacterial cells mostly contain a small structure of DNA double strand that is called plasmid, and that is distinct from the bacterial chromosome. So plasmids replicate independently from the bacterial chromosomes. Plasmids can be exchanged between the different bacteria without causing any harm. Moreover, their presence is not essential for the functioning of the eubacterial cell. However, the plasmid usually carries beneficial genes, such as genes for antimicrobial resistance, toxin production, resistance to the toxicity by toxic metals, and enzyme synthesis. So plasmids are the main structures that are used in biotechnological applications recently because they can replicate independently and can be inserted or removed easily from the bacterial cells and can be manipulated easily using different enzymes. Like eukaryotes, the prokaryotes have ribosomes for the synthesis of proteins, and however, they are structurally different in the number of ribosomal RNA and proteins they contain. And consequently, several antibiotics act by inhibiting the synthesis of proteins. by binding to bacterial ribosomes without affecting the host cell, such as the chloramophenicol and erythromycin. So next I am going to discuss about the Eubacteria versus Archaebacteria. Eubacteria versus Archaebacteria. So now the question is, what is the Archaea? Definition in biology. The Archaea are prokaryotic microorganisms that reproduce asexually by budding, binary fission, and fragmentation. They are highly specialized organisms that are known as ancient bacteria. So some Archaea live in extreme environments such as extremely high temperatures, referred to as thermophiles, while the others are Archaea that live in oxygen-free environments that are called methanogens. Halophiles are salt-loving Archaea growing in only environments rich in salt, such as the brines. Archaebacteria can also be found in some parts of the human body, such as the colon, mouth, and skin. Archaebacteria are not usually pathogenic. So now the question is, what is the difference between bacteria and eubacteria? Bacteria is a term that was previously used to include all bacteria. Soon two groups emerged: Eubacteria, true bacteria, and Archaebacteria or Archaea. So Eubacteria and Archaebacteria are the only prokaryotes that are found on Earth, and they have a common progenitor cell but different evolutionary lines. So the next question is, do bacteria have a nucleus? Both eubacterial and archaeal cells lack a nucleus and other membrane-bound organelles. Even though the archaea are structurally similar to bacteria, they are different when examined at a molecular level. For example, the bacterial cells usually have a peptidoglycan outer layer that varies in thickness in gram-negative or gram-positive bacteria. Next question is, but do archaea have the peptidoglycan? No. Some archaea, such as the methanogens, have a pseudopeptidoglycan S layer that forms a layer to resist the internal high osmotic pressure. Archaebacteria and eubacteria are both prokaryotic, though these organisms exist in different biological domains because they are so genetically different. It is thought that Archaebacteria are some of the oldest forms of the life on Earth, while Eubacteria arose later in evolutionary history. Eubacteria are now found in almost all environments, whereas Archaebacteria have been pushed to the only most extreme environments, such as the high salinity, lakes, thermal hot springs, and deep within the Earth's crust. There are a few other differences between these groups. For instance, the Archaebacteria are not susceptible to antibiotics, while the most eubacteria are unless they have developed the resistance. So Archaebacteria typically do not infect humans or aid in digestion, so this is generally not a problem for human health. Further, eubacteria have ability to form spores and survive the adverse conditions, and while Archaebacteria do not have this ability. Like eubacteria, Archaebacteria is a branch of prokaryotic evolution. However, unlike eubacteria, studies have shown them to be simpler and much ancient. For this reason, their cellular makeup is noticeably different from that of the eubacteria. For instance, the members of this group, Archaebacteria, lack peptidoglycan and contain the RNA polymerase and ribosomal proteins more similar to those of eukaryotes than eubacteria. Some examples of Archaebacteria including Pharoplasma, Halobacterium, Pyrobaculum, and Lochiarchium and Thermoproteus. So some of the species possesses a pseudopeptidoglycan. In these species, the N-acetylmuramic acid is replaced by the N-acetyltalosamine. uronic acid. For a majority of the species, the cell wall consists of a surface layer that is known as S layer. So it is made up of proteins and glycoprotein arranged in a hexagonal pattern and assembled into the lattices that range between 5 and 25 nanometer in thickness. Despite the difference in structure from that of Eubacteria, this composition serves to provide the mechanical support to the cell, in addition to their role in preventing the osmotic lysis. Apart from the differences in the cell wall composition, differences between Eubacteria and Archaebacteria are also evident in their cell membrane. Among Archaebacteria species, the hydrophobic lipid tails are bound to the glycerol through ether linkages, instead of the ester linkages present in the Eubacteria species. On the other hand, the fatty acid tails common in the Eubacteria are replaced by repeating units of 5 carbon that are linked in a manner allowing them to form tails that are noticeably longer than fatty acid tails. In cases where this produces a lipid monolayer, this provides an advantage to the hyperthermophiles, given that the structure prevents the membrane from peeling into two membranes of the domain. Archaea are divided into two phyla, and these include Archaeota. This group consists of extremophiles that are capable of surviving highly unfavorable conditions. A good example of these are methanogens, commonly found in environmental devoid of oxygen, and extreme halophiles that can be found in environments with the high salt concentrations. Next is the Archaeota. Organisms classified under this phylum are hyperthermophiles and normally found in hot aquatic environments, springs, and marine environments or surroundings. So some of the other characteristics of Archaebacteria include vary in shape morphology, for example, rod-shaped, spherical, or flattened, contains introns that are not coding sections of the non-coding sections of ribosomal RNA. Or RNA, and reproduces sexually through binary fission, fragmentation, and budding, and do not exhibit glycolysis. So next I am going to discuss about the main differences between Archaebacteria and Eubacteria. Eubacteria versus Archaebacteria. So first I will discuss about Eubacteria and then I will discuss about Archaebacteria. Eubacteria are complex microorganisms. Archaebacteria are simpler microorganisms. Eubacteria live anywhere on Earth, and Archaebacteria live mostly in extreme conditions. Eubacteria have peptidoglycan cell wall, but Archaebacteria have pseudopeptidoglycan cell wall. Eubacteria can obtain energy by the Krebs cycle or glycolysis, and Archaebacteria cannot perform the Krebs cycle or glycolysis. Eubacteria contains the L-glycerol phosphate in their membrane lipids, and Archaebacteria contains the D-glycerol phosphate in their membrane lipids. Eubacteria simple RNA polymerase, and Archaebacteria contains the complex RNA polymerase. Eubacteria contains the L-glycerol phosphate in their membrane lipids. Archaebacteria contains the complex RNA polymerase. Examples of the Eubacteria are Clostridium. Examples of Archaebacteria are Pyrobaculum. Further examples of Eubacteria are Bacillus and Pseudomonas and Mycobacterium. And further examples of Archaebacterium are Phaeroplasma, Lochiarchium, and Thermoproteus. So next I am going to discuss about the types of Eubacteria. The domain bacteria are classified according to the several characteristics including shape, Bacillus, Coccus, Spirochete, or Vibrio, the requirement of oxygen, facultative or obligate aerobes or anaerobes, nutrition, chemosynthetic or photosynthetic, and the composition of the cell wall, gram-positive or gram-negative. Eubacteria usually have one shape. However, in some cases, their shape becomes altered due to environmental conditions. Some Eubacteria are normally polymorphic, such as the such as the Corynebacterium and Rhizobium. And next are rounded Bacteria, may be elongated, flattened or oval. So after division and reproduction, they remain attached to each other. So Diplococcus means that the two cells remain attached to each other after reproduction, whereas the Streptococcus mean that they are attached to each other in a chain-like pattern. Those that remain in groups of 4 cells and divide into 2 planes are called as tetrads. So Staphylococcus are divided into different planes, forming the grape-like clusters or sheets. So bacilli bacteria can divide in one direction only so that they have a fewer forms of grouping than cocci. So bacilli can either be single bacilli, diplobacilli, pairs, or streptobacilli chains. So some bacilli are oval and look similar to cocci. Therefore, they are called as the Coccobacilli. So vibrio or spiral bacteria are twisted for one or more twists. So they look like the curved rods. Helical bacteria are called spirala and they have rigid bodies and look like a corkscrew. Eubacteria are classified into several phyla. Each bacterial phylum includes species characterized by specific features. Examples are as follows. So first is Proteobacteria. That include the most gram-negative bacteria. They are thought to have arisen from photosynthetic ancestors. Proteobacteria classified into 5 classes, and these are alpha Proteobacteria, beta Proteobacteria, gamma Proteobacteria, delta Proteobacteria, and epsilon Proteobacteria. So next is Cyanobacteria, are characterized by blue-green pigment. They perform photosynthesis as plants and algae, and many of these bacteria can fix nitrogen in the soil. Therefore, they are important in agricultural fields. So next is the Chlorobe. Phylum consists of photosynthetic bacteria. Members of this phylum are green sulfur bacteria. Chlorobe reduces carbon dioxide during photosynthesis using organic compounds such as carbohydrates and acids, and members of this phylum diverge greatly with their rod, spiral, cocci or budding forms. So next is Chloroflaxi. Members are green non-sulfur bacteria such as Chloroflaxi can perform photosynthesis. And next is the Chlamydiae. Chlamydiae members are pathogenic gram-negative cocci that have a unique cycle of development. They are transmitted from human to human by direct contact or respiratory airborne routes. So next is the Planctomycetes, are budding gram-negative bacteria. Even though their DNA is similar to bacteria, their cell wall is similar to Archaea. Additionally, some of them contain organelles similar to those of eukaryotes. And next is the Bacteroidetes. Bacteroidetes are anaerobic bacteria that inhabit the human intestinal tract or oral cavity or intestinal tract. So they are present in the feces and they may cause infection due to surgery or puncture wounds. And next is the Fusobacteria. Are anaerobic bacteria. Their cell shape is either paleomorphic or spindle. Next is the Spirochetes, are coiled resembling the metal springs and they are flagellated, and their flagella facilitate their movement using axial filaments. So Spirochetes are usually present in the human mouth, and bacteria commonly take on one of the 3 shapes like bacilli, cocci and spirala. So bacilli have a rod shape and cocci have a spherical shape, and spirala have a spiral or wave shape. Their shape was often used as a classification system until the recent DNA studies cast doubt on these classifications. So bacteria may stay linked after division, forming the other shapes such as clusters, filaments and tight coils. So Eubacteria are typically classified into gram-positive, gram-negative and miscellaneous. While there are many phyla of Eubacteria under the domain Bacteria, these relationships are often changing and are still being defined based on the new DNA experiments. So these are the shapes of the bacteria. Eubacteria can take on many different shapes. So first I will discuss about the shape cocci. Cocci has many different shapes like the diplococci. Diplococci is also known as the Tryptococcus Pneumoniae. This is the example of the diplococci. And the next is the tetrad. This is the tetrad shape. And the next is the staphylococci. And the example is the Staphylococcus aureus. And the next is the Sarcina. And the example is the Sarcina ventriculi. And the next shape is the Streptococci, and the example is the Streptococcus, and the example is the Streptococcus pyogenes. This is pyogenes, Streptococcus pyogenes. And the next shape is the bacilli. Bacilli, this is the chain of bacilli, and the example is the Bacillus anthracis. Next is the flagellate rods, and the example is Salmonella typhi. Next is spore former, and the example is the Clostridium botulinum, and the others are vibrios, and the example is the vibrio chlori, and next is spirilla, and the example is Helicobacter pylori, and these are the spirochetes, and the example is the Treponema pallidum. So all these are shown into the figures. Next is the further details about the types of Eubacteria. Some bacteria have an additional layer enclosing the cell wall, which is referred to as the bacterial outer membrane. This extra layer cannot be dyed with the Gram stain that is often used to classify bacteria by researchers. Thus, they are referred to as Gram negative, and bacteria that can be seen with a Gram stain are referred to as the Gram positive. So typically Gram negative bacteria contain more species that are pathogenic to humans, whereas Gram positive bacteria are either beneficial or not harmful to human health. So next I am going to discuss about evolution of the Eubacteria. So 3 domains of life was proposed in 1990s based on the fact that ribosomes are different in the 3 types of cells: Archaebacteria, Eubacteria, and Eukaryota. After comparing the nucleotide sequence in each cell, even though Eubacteria and Archaebacteria are prokaryotes, the 2 domains were separated due to variation in the small ribosomal RNA subunit in both domains. So Archaebacteria live in extreme environments, therefore they are thought to be the first organisms live on Earth. Molecular theories support the fact that the genes were transferred horizontally between the 3 types of cells, which consequently affected the evolutionary process of life. According to the endosymbiotic theory, some bacteria have evolved into mitochondria and chloroplasts by gene transmission. So accordingly, mitochondria and chloroplasts were genetically transferred between different domains to continue the evolutionary process. So this is the evolutionary tree of the 3 kingdoms as shown into the figure. So next I am going to discuss about the biological importance of Eubacteria. So the world is filled with the different eubacterial species, and our bodies contain different types and different species of eubacteria, which are biologically important in our life. Our bodies only of the eubacteria habitats forming our normal flora. So normal flora causes no harm to us and they are beneficial to our bodies. For example, they defend our bodies against pathogenic eubacteria, and others can produce the biologically important substances such as the B vitamins, as well as the vitamin K. So several species of eubacteria are used in the mass production of the chewable or tablet vitamins, since bacterial species provide an inexpensive, safe and non-toxic source of vitamins. For example, the Propionibacterium and Pseudomonas species produce the vitamin B12, whereas ascorbic acid vitamin C is produced by Cytobacter species using glucose. And next is the Streptomyces hygroscopicus is of particular importance in the pharmaceutical industry, since their different strains can produce about 200 different types of antibiotics. Eubacteria decompose organic matter and dead leaves into carbon dioxide and nutrients such as nitrogen, and they contribute to keeping the balance of all ecosystems. So next I am going to discuss about the eubacteria in ecology. Studying the relation between eubacteria and the environment is known as the microbial ecology. So it includes many branches that discuss how the eubacterial and other microorganisms interact with their environment. So eubacteria can convert the forms of oxygen, carbon, nitrogen and phosphorus that cannot be used by living organisms such as animals and plants into the useful forms. They fix the nitrogen found into the air and convert it into the soil. So moreover, they return the natural carbon dioxide into the atmosphere through the decomposition of dead plants and organic wastes. Carbon dioxide is then used by plants, cyanobacteria and algae to perform photosynthesis and obtain energy. So beneficial eubacterium can be used in sewage treatment. It converts the organic material and liquid harmful substances into organic unharmful substances such as carbon and nitrogen. Therefore, it helps in preserving the water by controlling pollution. So, Azospirillum is a bacterium that grows in the soil close to the roots of the plants. So it fixes the nitrogen from the air into the soil and uses the excreted nutrients from plants as a source of nutrition. So like Azospirillum, Rhizobium and Bradyrhizobium, which are commonly known as rhizobia, fix nitrogen in the roots of plants, especially the leguminous plants such as the peas and beans, and rhizobia are responsible for the formation of nodules in such plants. So next I am going to discuss about the examples of the fascinating eubacteria. So most people believe that bacteria are harmful organisms that causes diseases in humans, animals and plants, but actually only a few species of eubacteria are pathogenic. Many others are beneficial to all other living organisms, and eubacteria species are important in different fields such as medicine, agriculture, industry and energy production. In this section we are going to discuss the examples of fascinating bacteria. These are Nitrobacter and Nitrosomonas, are species of bacteria that have ability to use inorganic chemicals including carbon dioxide and energy sources as a source of carbon to produce complex chemicals which are reduced compounds of nitrogen. So Nitrosomonas can oxidize ammonium to nitrate to nitrite, whereas Nitrobacter can oxidize the nitrate Nitrobacter can oxidize the nitrite to nitrates in a process which is known as nitrification. The product of nitrification, nitrates, is a mobile form of nitrogen of great importance in the agricultural field. So, Zoogloia species contribute to the processes of treating the sewage such as the activated sludge system. While growing, these bacteria form a slimy and fluffy mess which is important in operating such systems. So Xanthomonas campestris can be ingested by humans. So it is capable of producing the xanthin using the lactose. So xanthin has a thickening effect. Therefore, it is used in the production of the salad, dressing, dairy products, shampoos, cold creams, and cosmetics. So Escherichia coli, abbreviated to E. coli, belongs to the Eubacteria domain, and it is classified into Proteobacteria phylum. It is rod-shaped and gram-negative, so it has an additional membrane surrounding its cell wall. So E. coli is commonly found in the gut of many different types of warm-blooded hosts, including humans, though it can become pathogenic under the right conditions. So most strains are harmless and possibly beneficial, but some can cause food poisoning and other illnesses. So the bacteria can only survive outside of a host for limited time. And next is the Tryptococcus Pneumoniae, which is abbreviated to the S. pneumoniae, is another form or another common eubacteria. It belongs to the Firmicutes phylum. So it has a spherical shape and it is gram positive. So S. pneumoniae can be found on healthy hosts in a respiratory tract, nasal cavity, and sinuses. However, the bacteria can become pathogenic and spread to the other parts of the body, often causing pneumonia. And next is the meningitis in immunocompromised hosts. The bacteria in high amounts can cause the other illnesses as well, including but not limited to the bronchitis, acute sinusitis, and sepsis. These examples both show that how the gram-positive versus gram-negative classification of pathogenic bacteria can be misleading. So many species of bacteria can be harmful or harmful under the different conditions. So next I am going to discuss about further details about eubacteria, so also referred to as true bacteria in some books. Eubacteria is a domain that is consisting of all the common groups of bacteria. As such, it's made up of all the species that fall within the bacteria domain. So while all the groups within this domain are prokaryotes, they display high diversity in their general morphologies, metabolism, and habitats. This has made possible to classify and describe different types of bacteria in nature. Various groups exist as parasites and are responsible for animal and plant diseases. However, the some are free-living, with some being beneficial to man. The eubacteria, also called just bacteria, are one of the three domains of the life, and along with Archaea and Eukarya, eubacteria are prokaryotic, meaning their cells do not have defined membrane-limited nuclei. As a group, they display an impressive range of biochemical diversity, and their numerous members are found in every habitat on Earth. So eubacteria are responsible for many human diseases, but also help maintain health and form vital parts of all the Earth's ecosystems. So some of the groups of bacteria that fall under the domain eubacteria include Proteobacteria, Cyanobacteria, gram-positive bacteria, and next is the spirochetes and chlamydias. So first I am going to discuss about the Proteobacteria. So Proteobacteria represent the largest and most diverse group among prokaryotes. As such, the division includes various parasitic forms, for example Salmonella, Heliobacter, and Escherichia coli, etc., as well as free-living forms such as those involved in nitrogen fixation. All members of the group are gram-negative and thus do not retain the primary stain during gram staining. Based on ribosomal RNA sequences, this division is divided into 6 major groups that include alpha, beta, delta, epsilon, gamma, and zeta proteobacteria. Based on metabolism, the division is also divided into 3 main subgroups that include the purple bacteria, photoautotroph and photoheterotroph that contain chlorophylls, and chemoautotrophic proteobacteria that is consisting of free-living and symbiotic members. Chemoheterotrophic bacteria, also known as enteric bacteria. So this is the scanning electron micrograph of the Helicobacter bacteria as shown into the figure. Next, I am going to discuss about the cyanobacteria, also known as blue-green algae. So cyanobacteria is a class of eubacteria consisting of photosynthetic microbes, which are commonly referred to as photosynthetic prokaryotes. These prokaryotes are ubiquitous in nature and can therefore be found in such environments as ponds, rivers, lakes, desert soil, and hot springs, among others. In these environments, some species are free-living given that they are capable of photosynthesis, or may form the symbiotic relationships with fungi, form the lichens. So some of the other characteristics associated with cyanobacteria include a thick and gelatinous cell wall, gliding among the motile species, and diverse morphological characteristics. So they may exist as single cells and colonies or as multicellular organisms where different cells can perform the different functions. So this is the unicellular bacteria form from a microbial mat in Guerrero, Negro, Baja California, as shown into the figure. Next, I am going to discuss about the spirochetes. As the name suggests, the spirochetes are characterized by a spiral morphology, and this architecture is the result of a flexible peptidoglycan cell wall which is covered by the axial fibrils. All the spirochetes are gram-negative and they tend to stain poorly, which makes it challenging to visualize using the routine microscopy. In nature, the spirochetes can be found in water bodies and decaying organic matter, animals, plants, and soil, etc. Like many other groups, the group consists of both free-living and pathogenic organisms that have been shown to cause the diseases in human beings and animals, for example, the Borrelia burgdorferi and Treponema pallidum. Some of the other characteristics associated with spirochetes include the covered by a bilayered membrane, and some species form the hyaluronic acid slime layer. Movement is made possible by the presence of an internal flagellar filament that causes spinning and flexing about the axis. So this is the Borrelia burgdorferi by the James Haney, Claudia Mullins, as shown into the figure. So next I am going to discuss about the chlamydias. Chlamydias is a group of organisms belonging to the family Chlamydiaceae, and they are obligate intracellular parasites of animals and are the most common causes of non-gonococcal urethritis in the United States. So Chlamydia trachomatis, on the other hand, is the most common cause of blindness across the globe. While the chlamydia species are grouped as gram-negative, their peptidoglycan is not easily detectable. Some of the other characteristics of chlamydia include they are immotile and are dimorphic in nature. So this is the transmission electron micrograph, shows the flower-like structure of the PMPD protein of the C. Trichomatis bacterium as shown into the figure. So next I am going to discuss about gram-positive bacteria. A majority of eubacteria are gram-positive, whereas some of the species in this group are capable of photosynthesis, which are called photosynthetic eubacteria. A majority are chemoheterotrophs and thus obtain nutrients by their surroundings. So being gram-positive bacteria, these species retain the primary color, which is the crystal violet dye during the gram staining, and thus appear violet in color when viewed under the microscope. This is because they contain a thick peptidoglycan layer compared to gram-negative species, and they vary in morphology from cocci species that are the spherical in shape For example, the Staphylococcus to rod-like species like bacilli such as the Listeria. Depending on the species, gram-positive bacteria can be found in different types of aquatic, marine, and freshwater and terrestrial environments. In the environment, the various species, for example, mycoplasmas and actinomycetes, have been shown to form endospore, while many of the species are parasitic, incapable of causing diseases in animals, and some of the species, for example, the actinomycetes, are beneficial in that they produce the medically relevant compounds such as antibiotics. So this is a 3D medical animation still showing the Staphylococcus bacteria as shown into the figure. Next, I am going to discuss about the structure. So like archaeans, the eubacteria are prokaryotes, meaning their cells do not have nuclei in which their DNA is stored. This distinguishes both groups from the eukaryotes, whose DNA is contained in a nucleus, and despite this structural resemblance, the eubacteria are not closely related to archaea, as shown by analysis of their RNA. So eubacteria are enclosed by a cell wall. This wall is made of cross-linked chains of peptidoglycan, a polymer that combines both amino acid and sugar chains. The network structure gives the wall the strength it needs to maintain its size and shape in the face of changing chemical and osmotic differences outside the cell. So penicillin and related antibiotics prevent the bacterial cell growth by inactivating an enzyme that builds the cell wall. So penicillin-resistant bacteria contain an enzyme that chemically modifies the penicillin, making it ineffective. Some types of bacteria have an additional layer outside the cell wall. This layer is made from the lipopolysaccharide LPS, a combination of lipids and sugars. There are several consequences to possessing this outer layer. Of least import to the bacteria but significant for the researchers is this layer prevents them from retaining a particular dye which is called gram stain that is used to classify bacteria. So bacteria that have this LPS layer are called gram negative in contrast to gram positive, which do not have an outer LPS layer and which do retain the stain. So of more importance to both bacteria and the organisms that in fact is that one portion of the LPS layer called the endotoxin is particularly toxic to humans and other animals. So endotoxin is partly to blame for the damage done by the infection from the salmonella and the other gram-negative species. Within the cell wall is the plasma membrane, which like the eukaryotic plasma membrane is a phospholipid bilayer and studded with the proteins. So embedded in the membrane and extending to the outside may be flagella, which are whip-like protein filaments. So powered by molecular motors at the base, these spin rapidly, propelling the bacterium through its environment. Within the plasma membrane is the bacterial cytoplasm. Unlike eukaryotes, the bacteria do not have any membrane-bound organelles such as mitochondria or chloroplasts. In fact, the two organelles are believed to be evolved from eubacteria that took up residence inside an ancestral eukaryote. So bacterial cells take on one of several common shapes which until recently were used as a basis of classification. So bacilli are rod-shaped, cocci are spherical, and spirally are spiral or wavy-shaped. After division, bacterial cells may remain linked and these form a variety of other shapes, from clusters to filaments to tight coils. So next I am going to discuss about the metabolism. Despite the lack of internal compartmentalization, bacterial metabolism is complex and is far more diverse than eukaryotic metabolism. Within the eubacteria, there are species that perform virtually every biochemical reaction which is known, and much bacterial chemistry remains to be discovered. Most of the vitamins humans require in our diet can be synthesized by bacteria, including the vitamin K. Humans absorb from the Escherichia coli, E. coli bacteria in our large intestines. The broadest and the most significant metabolic distinction among the eubacteria is based on the source of energy they use to power their metabolism. Like humans, many bacteria are heterotrophs, consuming organic carbon containing high energy compounds made by the other organisms. Other bacteria are chemolithotrophs, which use the inorganic high energy compounds such as hydrogen gas, ammonia, or hydrogen sulfide. And still others are phototrophs, using sunlight to turn the simple low energy compounds into high energy ones, and which they then consume internally. For all organisms, the extraction of energy from high energy compounds require a chemical reaction in which electrons move from atoms that bind them loosely to atoms that bind them tightly. The difference in binding energy is the profit available for powering the other cell processes. In almost all eukaryotes, the ultimate electron acceptor is oxygen, and water and carbon dioxide are the final waste products. Some bacteria use the oxygen for this purpose as well, and others use the sulfur-forming hydrogen sulfide, which has a strong odor. Carbon-forming flammable methane common in the zoams, and a variety of the other compounds. So bacteria that use oxygen are called aerobes, and those that do not are called anaerobes. And this distinction is not absolute. However, since many organisms can switch between the two modes of metabolism, and others can tolerate the presence of oxygen if they do not use it. So some bacteria die in oxygen, however, including the members of gram-positive Clostridium genus. Clostridium botulinum produces the botulinum toxin, that the deadliest substance known, and C. tetani produces the tetanus toxin that is responsible for tetanus and lockjaw, while the other Clostridium species are causing the gangrene. So next I am going to discuss about the life cycle. When provided with the adequate nutrients at a suitable temperature and pH, E. coli bacteria can double in number within 20 minutes. This is faster than most species grow, and faster than E. coli grows under natural conditions. Regardless of the rate, the growth of a bacterium involves synthesizing double the quantity of all its parts, including membrane, proteins, ribosomes, and DNA, and separation of daughter cells called binary fission is accomplished by creating a wall between the two halves. The new cells may eventually separate and may remain joined. When environmental conditions are harsh, some species, including members of the genus Clostridium, can form a special resistive structure within themselves, which is called an endospore. The endospore contains DNA, ribosomes, and other structures needed for life, but is metabolically inactive. It has a protective outer coat and very low water content, which help it survive heating, freezing, radiation, and chemical attack. So endospores are known to have survived for several thousand years and may be capable of surviving for much longer, possibly millions of years. When exposed to the right conditions, the presence of the warmth and nutrients, the endospore quickly undergoes the conversion back into an active bacterial cell. So next I am going to discuss about the DNA. DNA, the most eubacteria have DNA that is present in a single large circular chromosome. In addition, there may be numerous much smaller circles which are called plasmids. Plasmids usually carry one or a few genes. They have often specialized functions such as metabolism of a particular nutrient or antibiotic. Despite the absence of a nucleus, the chromosome is usually confined to a small region of the cell called the nucleoid and is attached to the inner membrane. The bacterial genome is smaller than that of a eukaryote. For example, E. coli has only about 4.6 million base pairs of DNA versus 3 billion in humans. As in eukaryotes, the DNA is tightly coiled to fit it into the cell. Unlike eukaryotes, however, the DNA is not attached to the histone proteins. So much of what we know about DNA replication has come from the study of bacteria, particularly E. coli, and the details of this process are discussed elsewhere in this encyclopedia. Unlike eukaryotic replication, prokaryotic replication begins at a single point and proceeds around the circle in both directions. The result is two circular chromosomes which are separated during cell division. So plasmids replicate by a similar process. Next I am going to discuss about the gene transfer. While bacteria do not have the sex-like multicellular organisms, there are several processes by which they obtain new genes, which are conjugation, transformation, and transduction. So conjugation can occur between the two appropriate bacterial strains when one or both extends hair-like projections called pili to contact each other. The chromosome or part of one may be transferred from one bacterium to the other. In addition, plasmids can be exchanged through these pili. So some bacteria can take up DNA from the environment, a process which is called transformation. The DNA can then be incorporated into the host chromosome. Some bacterial viruses called the phages can carry out the transduction. With some phages, the virus temporarily integrates into the host chromosome. When it releases itself, it may carry the some part of the host DNA with it. And when it goes on to infect another cell, this extra DNA may be left behind in the next round of the integration and release. Other phages, called the generalized transducers, package the fragments of the chromosome into a phage inserted instead of their own genome. When the transducing phage infects a new cell, they inject the bacterial DNA, and these phages lack their own genome and are unable to replicate in the new cell. The inserted bacterial DNA may recombine, join in with the host bacterial chromosome. So next I am going to discuss about gene regulation and protein synthesis. Gene expression in many bacteria is regulated through the existence of the operons. An operon is a cluster of genes whose protein products have related functions. For instance, the lac operon includes one gene that transports the lactose sugar into the cell and another that breaks it into two parts. These genes are under the control of the same promoter and so are transcribed and translated into protein at the same time. RNA polymerase can only reach the promoter if a repressor is not blocking it. The lac repressor is dislodged by lactose. In this way, the bacterium uses its resources to make the lactose digesting enzymes only when lactose is available. Other genes are expressed constantly at low levels. Their protein products are required for housekeeping functions, such as membrane synthesis and DNA repair. One such enzyme is DNA gyrase, which relieves strain in the double helix during replication and repair. DNA gyrase is the target for antibiotic, which is known as the ciprofloxacin, which is sold under the name of Cipro, effective against the Bacillus anthracis, the cause of the anthrax. Since the eukaryotes do not have this type of the gyrase, they are not harmed by the action of this antibiotic. As in eukaryotes, translation protein synthesis occurs on the ribosome without a nucleus to exclude it. The ribosome can attach to messenger RNA even while the RNA is still attached to the DNA. So multiple ribosomes can attach to the same messenger RNA, making multiple copies of the same protein. The ribosomes of Eubacteria are similar in structure to those in Eukaryotes and Archaea but differ in molecular detail. This has two important consequences. First, sequencing ribosomal RNA molecules is a useful tool for understanding the evolutionary diversification of Eubacteria. So organisms with more similar sequences are presumed to be more closely related. The same tool has been used to show that Archaea and Eubacteria are not closely related despite their outward similarities. Indeed, the Archaea are more closely related to eukaryotes, including the humans, than they are to Eubacteria. Second, the difference between the bacterial and eukaryotic ribosomes can be exploited in designing the antibacterial therapies. Various unique parts of the bacterial ribosome are the targets for the numerous antibiotics, including the streptomycin, tetracycline, and erythromycin. So next I am going to discuss about the size and shape. Prokaryotes range in size from 0.2 micrometers to more than 50 micrometers, although the average prokaryote is around 1 to 3 micrometers in size. Eukaryotic cells are approximately one order of the magnitude larger, ranging in size from 5 to 20 micrometers in diameter, with an average size of the 20 micrometers. The bacteria come in a number of distinct shapes as well. Common shapes include the spherical coccus, cylindrical rod, and spiral form spirula. While bacteria are generally regarded as unicellular organisms, they are also the examples of bacteria that exist as multicellular colonies, aggregates, or filaments. In addition to bacteria can aggregate on surfaces called biofilms, these assemblages can consist of a single species or communities of microorganisms that can participate in metabolic cooperation. Next is the origin of the bacteria. It is not known whether the ancestor of bacteria originated on Earth or elsewhere. Some scientists believe that a life form existed extraterrestrially in the Martian meteorite ALH 84001. Whether primitive life originated on Earth or elsewhere, current consensus is that the bacteria were present on Earth 3.8 billion years ago. So next I'm going to discuss about the diversity. Bacteria show an incredible range of metabolic diversity. So some bacteria can get their energy from light, and these are referred to as the phototrophic bacteria or phototrophic organisms. Organic compounds, organotrophic or inorganic compounds such as hydrogen sulfur compounds, inorganic nitrogen compounds, or ferrous ion compounds, chemolithotrophic. Some bacteria can make all of their organic compounds by fixing carbon, autotrophic, while others need to break down organic compounds to provide a carbon source, heterotrophic. So many bacteria are capable of fixing atmospheric nitrogen as the nitrogen source, in addition to organic and inorganic sources of nitrogen. Because of their metabolic diversity, bacteria play an important role in biogeochemical cycles, such as the carbon, nitrogen, and phosphorus cycles. This metabolic diversity also permits them to occupy a wide range of habitats. So bacteria can thrive in extreme soft temperatures, pH, salt, pressure, or toxic substances. Some bacteria can survive these conditions by spore formation, while other bacteria are able to multiply under the extreme conditions. The most primitive bacteria extant today are thermophiles, leading to the consensus view that life arose under extreme conditions. Within and between these extremes, the bacteria are found in marine, aquatic, terrestrial, and subterranean environments. There are bacteria that are obligate aerobes and some that are obligate anaerobes and many that fall somewhere in between. In recent years, highly conserved genes, such as the gene coding for the small subunit ribosomal RNA, have been used as principal taxonomic tractors. As bacteria evolve over time, the sequence of this molecule changes, allowing the taxonomic relationships between bacteria to be discerned. Many divisions exist within bacteria. An example of this diversity is the subdivision Alpha Proteobacteria, whose members are more diverse from each other than the plants from animals. More recently, full genome sequencing has revealed that genes move between the cells and even between species. Thus, bacterial genomes are in constant flux, driven by the gene acquisition from other species as well as evolutionary forces. The non-bacterial tree of life is remarkable, but as 99% of bacterial life remains uncultured, this tree will be undoubtedly expand greatly over time. So next I'm going to discuss about the associations. While the most bacteria are free-living at some point of their life cycles, many bacteria are capable of living in close associations with other organisms, including eukaryotes. Some of these so-called symbiotic associations are so highly evolved as to be obligate, while the other associations are facultative, meaning that symbiotic partners can live apart from each other. In some symbiosis, The eukaryotic host provides a highly specialized structure within which the bacteria reside, such as nitrogen-fixing root nodules found in leguminous plants such as the clover or the rumen, possessed by some herbivorous mammals. Looser symbiotic associations exist where the host provides no specialized structure for symbiotic bacteria. So organisms that populate the root zone of the plants can provide growth benefits. These bacteria are in turn making the use of plant products exudate through the roots. There are also the bacteria that are very harmful or even fatal to the eukaryotic host. An example of this is the Yersinia pestis. Yersinia pestis, which is a causative agent of the bubonic plague. Not all the associations between bacteria and eukaryotic hosts have such a drastic results. Many bacteria exist in relatively benign associations with their hosts, such as the Escherichia coli bacteria in the human large intestine. Some resident bacteria can become pathogenic under the certain circumstances. These opportunistic pathogens can cause the serious infection in the host whose defenses are compromised by the age or previous illnesses. Some association can be very intimate, occurring on an intracellular level. It is generally accepted that eukaryotic chloroplasts in mitochondria arose from the associations between bacteria and the other cells. These organelles are similar in size to bacteria and contain the remnants of the bacterial genomes. So next I'm going to discuss about the cellular structure. Bacteria cells are fairly simple in structure when compared to the eukaryotic cells of fungi, protists, plants and animals. As seen with an electron microscope, the majority of bacterial cell volume is filled with ribosomes, the sites of protein synthesis. Some bacteria, such as those that are photosynthetic, contain many internal membranes where the metabolic processes takes place. They contain no internal organelles such as the mitochondria and chloroplasts. Like Archaebacteria have a prokaryotic cell organization. Their deoxyribonucleic acid DNA is loosely gathered into a nucleoid and is not surrounded by a nuclear membrane like that found in eukaryotes. The DNA usually occurs as a single long circular strand, but some bacteria have linear chromosomes or divide their genetic material into several DNA molecules. Although they are different from the chromosomes of eukaryotes, the large circular or linear prokaryotic DNA molecules are often termed as chromosomes as well. Bacteria can also have smaller circles of the DNA, which are called plasmids, which usually carry a small number of genes used for specific metabolic functions. For example, to allow the bacteria to metabolize the certain compounds. So plasmids can easily be passed from cell to cell, allowing the bacteria to rapidly pick up new metabolic functions and are basis for many advances in genetic engineering. Like all the living cells, bacteria are surrounded by lipid membranes, so most bacteria also have cell walls made up of the peptidoglycan called murin. The peptidoglycan layer is made up of a single kind of molecule from covalently linked sugar derivatives and amino acids. This molecule surrounds the bacteria cell like chain mail armor. Together with the osmotic pressure, the wall gives cells rigidity and shape. The cell wall structure in the presence or absence of a second lipid membrane surrounding the murin layer determine how the bacteria react in a procedure called the Gram stain. So gram-positive organisms which take up Gram stain have a single membrane and very thick outer peptidoglycan layer. The Gram-negative organisms do not take up the stain and have two membranes in between, which is a thin layer of peptidoglycan. Next I am going to discuss about distribution and ecological roles. Although the bacteria may appear simple, They excel in the diversity and complexity of their metabolic capabilities, and they are able to survive in many places. So bacteria are found everywhere on earth where the life is able to exist. They are plentiful in soils, bodies of water, on ice and snow, and are even found deep within the earth's crust. They often take advantage of living in and on the other organisms in symbiotic relationships, and can be found in inhabiting the intestinal tracts and surfaces of animals, including humans. For the most part, the bacteria in and around us bring us more benefit than harm. Sometimes, however, bacteria can be pathogenic or disease-causing. This can happen for a number of reasons, such as when the host has compromised immune system, or when a bacterium acquires the genes that make it grow more aggressively or secrete the toxins into its host environment. Oxygen producing photosynthesis, which is so familiar in plants, is actually a bacterial invention. Many bacteria are photosynthetic and use the light energy to turn the CO2 from the atmosphere into the cell material. Among these, only the Cyanobacteria produce oxygen during photosynthesis. Plastids, the photosynthetic organelles found in plants and algae, evolved from the Cyanobacteria through a process which is called endosymbiosis, in which the Cyanobacteria lived inside the cells of other organisms that were ancestors of the green algae. And mitochondria found in most eukaryotic cells also evolved from the non-photosynthetic respiring bacteria in this way. So bacteria are crucial for the cycling of elements necessary for all life. Through various processes which we generally call decomposition, so bacteria break down the cell materials of dead organisms into simpler carbon, phosphorus, sulfur, and nitrogen containing nutrients that can be used again by the other organisms for growth. Without bacteria to recycle these essential nutrients, they would remain within the dead organisms or sediments and would thus be unavailable for use by the other organisms. If you like my video, please subscribe to my YouTube channel and press the bell icon as well for further notifications and updates. Thanks for watching. Thank you so much for your time and appreciation.